Hi, I'm Natasha Gurevich. I'm a second-year bioinformatics student in the Campbell Lab at Boston University.
Hi, I'm Jocelyn Velasquez Reyes, and I'm also a second year PhD student at Boston University in the Connor Lab. We have a third member of this team that has been working on this project, Helen Scott, who is also a second year in the Segre Lab at Boston University.
Scientists have been studying and culturing bacteria for several decades now. Our ability to culture bacteria has led to incredible discoveries in various branches of science, including medicine and evolution. The versatility in the scientific community has led to several repositories to make these model organisms accessible. But does this accessibility lead to laboratory domestication? Laboratory domestication occurs when the culturing of bacteria leads to the organism diverging from the original wild-type sample. Long-term propagation of a bacterial strain can accumulate mutations over time so that a lab may unknowingly develop a cultured strain distinct from the wild ancestor. As mutations often occur randomly, two labs may develop different cultivated strains for the same wild ancestor. This could potentially decrease the reliability of experimental results across labs. It is therefore critical to perform careful and extremely short-term propagations of natural isolates to conserve the traits encoded in their original genomes and limit the creation of cultivated strains. To understand how bacterial stocks could be changing due to the selective pressures that arise in a lab environment, we resequenced 59 marine bacteria and compared them to reference genomes collected prior. We then sought to understand the factors affecting mutation profiles, factors affecting mutation rates, and functional genomic changes.
So to start off, we're going to begin by talking about factors impacting mutational rates. And we wanted to consider the time between the reference, the resequenced genome, the ratio of SNP count versus genome length, as well as the type of repository that the bacteria is coming from. To elaborate, was it coming from an independent academic lab, or was it coming from a known culture repository like ATCC? We hypothesized that more time between the reference and the resequenced genome should result in a greater SNP count. Additionally, we can expect that there is less change in strains coming from culture repositories like ATCC than smaller non-specialized labs. The analysis to answer these questions is right here in the form of a scatter plot. The x-axis is the year that the reference genome was first sequenced. The y-axis is the ratio normalized of SNP count versus genome length, and the different colored dots are correlated to where the bacterial stock is coming from. And as you can see, there is no clear trend suggesting that these factors don't actually affect mutation rate. And to answer and address our other analysis, the functional impact analysis, which is right next to it here, we started off by looking broadly on the scatter plot, but then we wanted to zoom in and actually look at what regions of the genomes these SNPs are coming from. So to do that, we ended up doing genomic annotation on our VCFs to find how many SNPs were associated with a given gene. And what we quickly found out, which you can see in these bar plots right here, is that the samples had really unique mutations affecting specific genes. So that's very interesting because that means that there is no shared affecting all the bacteria. But then we also wanted to know, okay, well, if we zoom in a little bit more, are there some genes that actually are shared across some samples? And we ended up seeing that RPOB and RPOC are really shared, mutations shared across several samples of different backgrounds, which is very interesting. And additionally, those genes are associated with the subunit RNA polymerase, which is often a suggestion of selective pressures that make these mutations arise.
We also sought to understand which factors affect the mutation profiles of the samples. We defined a mutation profile for a sample here as a vector of 12 entries containing counts of the 12 different single base substitutions. Using these mutation count vectors, we used hierarchical clustering to create this dendrogram you see here that groups samples based on how similar their mutation profiles are. We could then observe if there were any common factors between samples that were grouped closely. From the color bars here on the dendrogram, we can see that neither length of in-lab propagation, which is labeled as year here, nor genome length have an impact on the mutation profile. GC content does appear to have some influence. The samples on this major branch of the tree tend to skew towards the higher range of the GC content scale, while those down here tend to skew toward the lower range. To observe the effect of phylogeny on mutation profiles, we constructed a phylogenetic tree of the reference genomes, which we compared to the hierarchical clustering tree from the mutation profiles. We searched for subgroups on each tree with common membership. The two tanglegrams that you see here, we have blue labeled samples, which representing those samples that are common in the two subtrees being compared, while the samples that are labeled in red are unique to one subtree or the other. Both trees have an initial branch point that splits the tree into two major subgroups. One subgroup on the mutation profile tree over here correlates pretty well to a clustered portion of one of the major phylogenetic tree subgroups over here. The remaining samples in that phylogenetic tree subgroup down here correlate to the other major subgroup of the mutation profile tree over here. The second major subgroup of the phylogenetic tree, which is over here, this correlates mostly to the mismatches from the previous comparisons.
So with all of our research results shared, we wanted to conclude this by saying that resequencing bacterial strains after propagation in lab and evaluating the difference from their reference genomes, we found that genome size, length of lab propagation, type of culture repository did not influence mutation rate. And the main genes shared across all the samples seem to be RPOB and RPOC. And they were the most frequently mutated. The estimated time of lab propagation and mode of sample storage did not correlate with mutational patterns, while phylogenetic relationships among samples and GC content did. These results provide a further understanding of what factors influence bacterial mutation within the laboratory, as well as which types of mutation are commonly occurring.